Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Overcoming Challenges in Pediatric Clinical Trials with Digital Devices. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, webinars are designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can also contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Obvio Health, who developed the content for this presentation. Obvio Health is an end-to-end virtual research organization leading the digitization of health innovation in the pediatric space. Since 2017, they've been pioneering decentralized clinical trials with a patient-centric platform, an in-house virtual site team, and expert clinicians, all dedicated to tackling inefficiencies at every touchpoint for the clinical trial process. They have completed 49 hybrid and fully decentralized clinical trials, 11 of which were in the pediatric space. Now I would like to introduce our speakers for today's event. For over 25 years, Susan Delabrita, PhD, has been a leader in clinical trials healthcare. An expert in clinical innovation, she specializes in rater and patient training, real-world data collection in decentralized clinical trials, as well as ePro, ClinRo, and instrument development and validation. Jaya Punati, MD, is a board-certified pediatric gastroenterologist with multiple fellowships in pediatric gastroenterology, neurogastroenterology, and motility. She currently serves in several roles at the Children's Hospital of Los Angeles, where she specializes in pediatric colorectal and gastrointestinal motility disorders. And Brian Cohn, PhD, is a computational biologist specializing in development and application of creative digital solutions in healthcare and clinical studies. Dr. Cohn's expertise includes the study of human biomechanics and AI-driven digital instruments. He is the Director of Research at Obvio Health, leading research teams in pediatric outcomes devices. And Holly Link, PhD, has devoted her career to pragmatic applications of molecular biology in the field of healthcare, primarily in pediatric nutrition, illness prevention, and enigmatic virus diagnostics. These applications range from exploiting the modification of expressed human genes for large-scale therapeutic proteins to nutrient-based innovation against common infant feeding dilemmas. Now, without further ado, though, I'd like to go ahead and hand over the mic to our speaker today, Holly Link. You may begin.
Thank you. Thank you very much, Ryan. I'm really excited to have you all here. As Ryan said, my name is Holly Link, and I'm the moderator today. As you might infer, this webinar seeks for us to share a few of the tools that have been developed by Obvio Health and our partners for site-independent data collection. Specifically, we have pediatric studies in mind, and these tools can lessen the burdens of both the caregivers or the participants, as well as the people conducting or providing the study. Our three panelists are going to share some experiences and some examples of digital solutions that are particularly suited for frequent evaluations within pediatric trials. So we hope that these presentations will generate a lot of questions on your part, and we encourage you to share any perceptions that you might have or offer any particular problems that you think would be really well suited to be solved by these digital tools. So please use the Q&A section on your screen for that purpose. So questions or comments can pop up there. So with that, we can move ahead and take a quick look at the agenda. We've done the introductions, and now we're going to start with our very first speaker, Susan Delabrita. She's going to be discussing barriers to pediatric pro in clinical research. We will follow that with Jaya discussing patient reporting challenges, a clinical practice perspective, and wrap things up with Brian covering the promise of digital tools to augment ePro. And hopefully we'll have plenty of time left for your Q&A. So let's turn it over now to Susan, please. Oh, I forgot, polling question. My mistake.
That's all right. Yes, so we've got a polling question for audiences. This should be appearing on everyone's screen right now. You can participate in this poll by selecting on any of the answers that you see in front of you and then clicking submit. The question that we're asking you is, while thinking about the pediatric studies that your company conducts, what aspects of data collection do you find most challenging? Your answer options are maladapted instruments or tools for pediatric patients, challenge of capturing and evaluating real-time data, inability of pediatric patients to self-report symptoms, caregiver subjectivity in reporting, or not applicable to my organization. We'll give everyone a few seconds to consider their answer as it best applies to the question that we're asking, which again is thinking about the pediatric studies your company conducts. What aspects of data collection do you find most challenging? Let's go ahead and take a look at the results. It looks like we have here 31% of you have selected caregiver subjectivity and reporting, as well as 31% selecting challenges of capturing and evaluating real-time data. Next to that would be 15% at maladapted instruments or tools for pediatric patients, and then 12% for each for inability of pediatric patients for self-reporting symptoms or not applicable to our organization. It's back to you, Holly.
Okay, well, I see we have a tie here between capturing and evaluating real-time data and caregiver concerns. So we will go to Susan now.
Thank you very much, Holly. Good morning, good afternoon, good evening. It's a pleasure to be here with all of you today. We really appreciate you coming out to the webinar and looking forward to discussing with you many of the challenges that are inherent in doing pediatric clinical trials and doing them well, and also how we have developed creative solutions, both in terms of our strategy and our approach, the use of technology, digitization, as well as innovations within this space to address some of those challenges that just came up in the previous polling question. If we advance to the next slide, please. So this is not intended to be a comprehensive list of all of the issues that are specific to pediatric clinical trials, but I've been working in clinical trials now for over 28 years, and myself alone have done hundreds of pediatric clinical trials, including many in rare disease, where the stakes are even higher, so to speak, in terms of all of these issues that compromise the signal-to-noise ratio, make it harder to see efficacy and safety endpoints. And I think Holly talked about this, early on, and so did Ryan, which is, my specialization is really in outcomes optimization, data quality, optimizing the signal-to-noise ratio, and making sure that we avoid false positive and false negative outcomes within a clinical trials data set. And this is just a list of some of the top challenges that I've seen over the years in doing these, is that, when we're doing pediatric trials, very often the trial is from ages 4 to 17 or 18. And as soon as you see that, there's some inherent challenges, and where is the cutoff in terms of self-report versus caregiver report, knowing that there is a great value in self-report, and it is most critical that everyone that can self-report does self-report. And the reality is, a simple cutoff is not the solution. It really depends on the concept, and it also depends on the cognitive abilities of the child in the study. And one of the mechanisms we've used to address this is using training and quizzing to make sure we can use self-report cutoff at a level that is more granular than doing a simple age cutoff. But at the end of the day, you've got to develop different instruments, some that are self-completion and some that are caregiver completion. And as you know, there's not a lot of great pediatric instruments out there. Those old smiley face charts are just bad. I can tell you, not only do adults not understand them, children have no idea what all those faces mean either. So there's a lot of things we've been doing as an industry over the year where we've made assumptions, we used what we could, we grabbed hammers to solve problems that hammers were not meant to solve. And what we're here to talk about today is applying precision instrumentation, better ePro, better innovations, capture of images and audio, and Brian and Jay are going to talk about this later about how do we break through this feeling of where we've been stuck for several years now. And then the other layer of complexity when we think about reporting is in these younger demographics that are incapable of self-report, we have real challenges with the caregivers for a number of reasons. One, caregivers are estimated to spend over 40 hours a week in the role as a caregiver. So typically we're talking about mom or dad or a close family member where you've got emotional ties as well as the burden of having to deal with the child in your care. And in many cases, knowing this, a clinical trial may be the last hope for that child. So it's very common for caregivers and parents to inflate ratings at baseline out of despair. And as soon as the child is enrolled in the study, those ratings come down. It has nothing to do with intervention or therapeutic progression or regression. And then when we further compound that, if you have more than one caregiver, you can have two caregivers that look at the exact same, child is in pain. Caregiver A may say, that's not so bad, that's a 2. And caregiver A may say, oh my goodness, my child, and give that an 8. And so you have all of this subjectivity that's compounding it. And then you've got a third layer, if you're using sites in your clinical trials, where now you've got not only a dyad between trying to report on neutral observable behaviors, which is the spirit of the clinical trial, to untie that from the emotional tolls associated with being a caregiver and seeing a child in your care who is ill, to transmission of that data potentially to a clinician at the site. And so the dyad becomes a triad of complexity. And at the end of the day, what we're looking for is observable behavior versus projecting or emotional attachment. And so we definitely utilize training as one of our major mechanisms to help explain that not only do the clinicians, but the caregivers themselves need to dissociate from their normal roles and become a neutral observer and reporter in a clinical trial. So you see a lot of placebo response rates that are very high in these trials. And like I said, we have learned that training is one of the solutions for that. We also know that, like I said, a lot of the tools out there are hammers. They are just hammers. And Jaya and Brian and I are here to talk to you today about developing much better tools. There are a lot of places where we can either put a better tool in the hands of a caregiver, or we can take that burden off of the caregiver and say, let us do it. Get us a photo, get us a video, get us something, and we'll take it from there. And what you'll see as we progress through today's talk is it greatly improves the sensitivity of the outcomes we're able to get from this data. Next slide, please. One of the things that I just want to make sure we level set on is that this is something that has been very well tested over the years. Adults prefer digital data capture, and children just as much, if not even more so. Any study that's been done and any therapeutic, we put 2 up here, but we could have put 222 up here. The reality is, and I think we all know this, if you have a child or you know a child, iPads, smartphones, it's hard to pry them out of their little hands. And if you do, you're very lucky. They really do appreciate digitization, gamification, and they would much prefer using digital devices than using paper or traditional methodologies. Next slide, please. The other thing that I'd like to talk about here is about there are places where humans, whether it's a child or an adult, just struggle with reporting accurately on a data set. And one of the things I've worked on for many years is both adult and pediatric diabetes clinical trials. And there are several studies and meta-analysis that show what they did, and this is a very interesting study about humans, is if you give a human a blood glucose meter and you ask them to do a finger stick read, and then you ask them to report that on paper, and you don't tell them that the blood glucose meter is recording all of the actual values, so they have no idea. So you end up with two data sets, actual, the real blood glucose readings, and what's recorded in a paper diary or even transmitted to an electronic diary. What happens is you end up with two very different data sets, and there are three fundamental problems with that data set that's not coming directly from the meter. One is something we call phantom values. And what that means is the meter has no values, but the patient is reporting a date and a time and a value. So you know that's out. And then there are sometimes values on the meter, but they don't appear in the record, so they don't make it. And then the third category of errors, which is something we all struggle with, is trying to be normal. Diabetes patients very much struggle to report excursions, both high and low. And so what that means, if I measure my blood glucose and I get a 200, I am very likely to report it's about 130. And you see the meter says it's 200, and the other patient source says it's 130. So for many years, we've been capturing data that is just fundamentally incorrect and trying to draw conclusions about efficacy and safety of various drugs and devices. And it just isn't a good approach. And the long and short of this is, as bad as this, what we now know that about 50% of this data set in adults is all of the things I just discussed. Phantom values, values that are pushed toward the norm, and so on. This problem is exacerbated in adults and the teenage population. It gets even worse. But the good news is we can take all of this problems off the table with the things we're going to be talking about today, because we now have devices and Bluetooth and integration, and we can bring in those data sets, and we don't have to rely on patient transcription of this data anymore so that you can get to accuracy in your outcomes. Next slide, please. And here, this is just one example, expanding on what I was talking about earlier, is engagement, gamification, anything you can do to make something more interactive, more appealing, whether it's through imagery, whether it's through specific rewards. All of these things very effectively boost compliance in pediatric studies. Next slide, please. And this is something that I know Brian and Jay are going to talk a lot about today, but I want to set up the fundamental problem. These are studies that we have done in-house and that we have talked about both at DIA and ISPOR. And that is, when we're talking about pediatric GI, we all know that the primary endpoint in a pediatric GI study, part of that endpoint will almost always be based on stool consistency. Stool consistency is not easy for caregivers to report, even in the best of settings. You have a lot of problems with it in terms of, one, if it's a child in a diaper, you're trying to sift through and sort of recreate what happened to the stool in a squish diaper. If things are going into the toilet, now you're dealing with the water and the timing, and there's just no good way to do this. And then on top of all of that, you have fundamental issue of caregiver subjectivity. Do you know the #1 most common used instrument in the world is the Bristol stool scale? 1, 2, 3, 4, 5, 6, 7 grades of stool, with 7 being complete water and 1 being as hard of the stool as can possibly be. And what I would like to show you in this data set is what we did is through the Avio Health app, we asked caregivers to send us images of stool and rate them. So like the diabetes example, we have 2 data sets. We have a data set of the images of the stool, and we have a central expert, a pediatric gastro expert, just like Jaya, who's taking a look at that image and providing a rating. And then we also have the caregiver rating. So everything you see in gray is a caregiver, and everything you see in pink is a central expert, just like Jaya. And what we saw is when we took a look at those images, we saw that caregivers had a very hard time on a number of these points. What you'll see is, just like the diabetes example, they tended to over-report normal. So if you look in the middle of the scale where stools would be normal, you see spikes in those gray bars. So they're pushing things more toward normal. If you look, they're having a hard time with the extremes, just like the diabetes example. You're not seeing super hard ones and twos. There's either no gray bar or a smaller gray bar. And then they struggle with those intermediate consistencies. I think we all do. This is a tough scale to use, but the experts don't. And then when we get to the sevens, complete water, it's almost like they just give up and push everything to either normal or water. And everything else in between is a real struggle. So we said, you know, there's definitely a better way to do this. This is a case where we can take burden off of the hands of the caregivers, and we can handle it with technologies like Brian is going to talk about and show you how we can reliably bring in those data sets, and how Jay is going to talk about how people like her and her teams have the capacities to score and rate these images and improve signal accuracy. And these are just some of the things that we're going to talk about today, but it sets the stage for how we're solving problems and changing the future.
Thank you, Susan. That was really educational, and I'm sure the participants not only appreciate your summary there, but they've been experiencing some of these issues as well in their own studies or studies that they would like to conduct, or even in their patient populations. And so, speaking of patient populations, I'm going to take us next to Dr. Punati. Jay is going to talk about patient reporting challenges, a clinical practice perspective, and marry together these two things, what's happening in clinical trials and what's happening in pediatric offices. So Jaya, to you, please.
Thank you, Susan. Thank you, Holly. This is very nicely described by Susan, the practical problems that translate into practicing medicine. Susan had talked about the Bristol stool chart, which is on the left side here. This is the chart that's being used very often in every pediatrician's office to assess the health of stool. Even if we try to make it fun to use your chocolate as various forms of stool, it is very, very hard to have parents, children look at the stool and be able to describe it accurately. Who wants to look at poop, at their bowel movements? Nobody. And as well as you may not be in a convenient place to spend a lot of time examining the stool to give accurate reports. So you have to rely on recall. And as Susan's data points, recall is good at the extremes, but not in between. So that tends to make it very hard to assess what's happening in the clinical setting. You add to that diapers, where a child may be moving around, rolling around, and by the time a caregiver actually opens the diaper to look at what the stool looks like or what the color of the stool looks like, the reported information is all over the place. The other point is having different caregivers exchange that information with each other so that it's consolidated and brought to the clinician. And then it's a free-for-all. It's even harder for us to assess what's going on. This is not a problem just at home. Even in the hospital, you have 3 nurses caring for the patient, giving us different reports and scoring. When adding stool color to the mix, there is a condition, biliary atresia, which is a life-threatening congenital disorder of the liver and can easily be diagnosed based on stool color. It is imperative it be diagnosed within the first 3 months of life because the treatment window rapidly disappears after that, and the child may end up requiring a liver transplant. Cards have been developed. Newborn parents are sent home with these cards to check it against the stool of the child every time. How many of us can find our things when we have a newborn baby at home. The parent is already stressed out about having a new child. There are multiple caregivers and different people who might be giving advice to the parent on what's normal, what's not, and recording all of this data and bringing it to the hospital at the clinic visits. Very difficult. So it would be nice to have tools that take the stress away from the parent. It would be nice to have AI-driven photographs or apps that all the parent has to do is click on the app and point it at the stool, and everything else is off their plate. The data gets transmitted to us, and we can, as providers, look at it. The other aspect is also getting consistency among different providers who look at these stools. Being able to say the same thing over and over for all provider care ratings. Here we have some examples of different stools, different types of stools. I'm going to focus on the one in the middle, which is the darker colored stool and is named meconium. And right at the very end is actually a stool that is a large amount of blood in that stool and is called melena. Melena is a life-threatening bleeding disorder that happens from large ulcers in the GI tract. Now, if a parent can tell the difference between a meconium or a melena, it can lead to either overcalling it and unnecessary tests in the child, really missing the boat and having the child get more ill before things are addressed. And then if you look at conditions such as milk protein intolerance, where there is a variable presentation of either a few drops or streaks of blood, which you can see on the second to last picture, or a couple of very tiny specks that a parent may not even notice. When we change the formula and we are trying to engage the family in looking at the diapers and giving us the information in clinic to see if we need to change formulas to a whole spectrum of partially broken down to fully broken down, then we run into trouble at data gathering. Colon cancer is also something that is screened just by looking at blood in stool. Next slide, please. So here's a picture, as we were talking about the amount of blood in the diaper. In the top picture, there is some blood clearly visible. In the bottom picture, you really have to look at the diaper closely to get that information. Right now, the best way, standardized way to look at the presence of blood is to do a fecal immunochemical test, which is collecting the stool and sending it to the lab. It's very hard for us to collect stool from a diaper, enough to be able to test. And there's a delay in the time it takes for turnaround. In the past, hemocal tests, which are little cards where you can swab a little bit of stool onto a stick and place it on this card and look for a color change, was used. It had to be, it's no longer popular and is out of favor because it's hard for people to determine what the color is. If there are people who have looked at pregnancy tests, you don't know whether you're positive or negative. Are you partially pregnant? Not going to happen, right? So the same way, it's really hard to have an idea of how much blood or is there blood just by looking at the color card with the naked eye. Now, if we had an AI system or a photo that could look at the color density and score it, that would be perfect and take subjectivity out of it. We also coming to the child that cannot verbalize their discomfort. As Susan had mentioned prior, we use the face scales. We try to attribute certain output to these faces, but it's notoriously unreliable. On the left, as you can see, there's so many expressions that we can give to this, but is it discomfort? Is it new food that the child is trying, or is that belly pain? And if you have an older child who cognitively is unable to use words to express their discomfort or their pleasure, that makes it even harder. So having tools which can give us what the set standards for the child, what the baseline utterances are, what is the standard comparable to children of the same age, babies of the same age, versus what's the baseline for that baby, and when it changes out of the baseline, then we can record concern. For example, in colic, how we have criteria that say that child may be fussy for 3 hours or more per day, but how the recall from caregivers about really, is it 3 hours? Is it 3 minutes? Is it longer than that? Is it between different caregivers and we haven't exchanged the time of fussiness? So all this makes it very hard to assess what's going on without and can easily be overcome if we had tools that could standardize this information. Next slide, please. It's not just in gastroenterology. There are other conditions that we can leverage AI. For example, in asthma control, parents are relying. We try to teach the parents to learn how to listen to the child's lungs and give them treatments. It's hard. I mean, there's variability in providers who are trained for several years on how they are able to listen to breath sounds, and it's even more harder for a parent to learn that. So if there was a tool that providers can just put it over the child's lungs and get an understanding of what kind of breath sounds are there and get a cue on when the inhaler is needed or not needed, and any breakthrough uses of these inhalers, that data can be sent to the provider. And we can look for improved management and home-based management without expensive hospitalizations and clinic visits to address this. So applications are endless. Next slide, please. Think about rashes. The children, one of the most common conditions in children is development of rashes as they're exposed to various environmental factors and dietary factors. As a clinician, it is difficult to be able to see all the types of presentations for rashes in your training. If the child's skin color is different, the same rash may appear, may not have the same textbook characteristics that the trainees are taught or the practitioners are taught. Having a database of all types of presentation and an AI app that you can point and it can sort through all of these images consistently and give a more accurate diagnostic data would help the provider and reassure the parents on simple conditions that don't really require a physician input. And that may just be transient. In clinical practice, really having better tools is invaluable.
It is indeed, Jaya. Thank you so much for sharing these areas that cross over between pediatric practice and pediatric trials. And we want to go on in a little bit with some solutions to that. But first, let me send you back to Ryan, who will deal with polling question number 2. And also give you the opportunity, remind you of the opportunity to put your questions in the little question box. Thank you.
Yes, thank you very much, Holly. So for our next poll question, again, attendees can participate by selecting on any of the answers you see in front of you and clicking submit. The question we're asking this time would like to know, where do you think digitization of data collection and similar remote activities would be the most helpful to you? Your answer options are pediatric patient and/or caregiver recruitment, accurate patient-caregiver reporting, real-time tracking of patient symptoms, or expedited paths to data analysis. We'll give everyone a few more seconds to consider their answer to this question as it best applies to yourself or your organization. Where do you think digitization of data collection and similar remote activities would be the most helpful to you? It looks like we have most of our votes in. Let's go ahead and see where your results have come out. We have 44% for accurate patient reporting,
then 28% for real-time tracking of symptoms, 24% then for expedited path to data analysis, and right at the end, 4% of you have selected pediatric patient and or caregiver recruitment. Thanks for your participation. Holly, it's back to you.
Thanks. So now we can move to our final panelist, Dr. Brian Cohn. He's going to give us the promise of digital tools for augmenting ePro. Brian?
Great, thank you so much, Holly. So really what I'm hearing through Susan's explanation of the challenges of the ePro and Jaya's challenges with delivering clinical tools at the actual clinical case, I think there's a lot of opportunities that we can talk about around many different parts of that life cycle. So let me just show you the next slide that shows a couple places where we can inject innovation. So if you want to augment ePro, you have to evaluate its parts and invent creative solutions for each step along the way. So from observation on the left, the way the participant collects that observation, where the pictures they're taking, the notes that are being made, the way we're notifying the participant to make observations, all the way to the way it gets saved in a database, checked if there's a problem or anomaly detection, or even as it finally makes it all the way to the outcome vault. Is it accurate? Are there ways that we can check and make sure that the data that ends at the end of a clinical trial is as accurate as possible? Every single one of these stages is an opportunity. So for today, I'm just going to be focusing on the first line, from observation to patient. So in this case, of course, you could increase the training, and that alone has made an incredible difference in a lot of the trials that we've run. Just creating better training materials, videos for the participant to watch instead of just read a giant paragraph. And those things have already made a huge difference. But today I'm going to be talking about AI-assisted ePro. And when I use AI, I'll be specific about the machine learning algorithms that we're using and training. But when we say augmented ePro, we refer to this kind of idea that in order to collect the next level type of patient-reported outcomes, we are collaborating with the parent. We are teaching the parent and guiding them, and also taking observations directly from the devices they already have and also new devices. So the three main examples I'll talk about today are, first, research question: how many minutes did your baby cry today? The second research question is, how many times did you cough today, or your child cough today? And finally, how has the skin condition changed over the past week? And how can we create better evidence for those three questions? And you can see that for each of those questions, that's a question we ask in many clinical trials. So first, let's hop into audio, the next slide. You can learn a lot about a child's condition by just listening. Of course, we use our eyes and our ears for everything and many other things, but we want to use audio as a way to generate clinical insight, from measuring coughs and sneezes to breathing patterns, and understand across both adults and children, how many times do they sneeze? What types of hiccups are we seeing? Can we detect vomits and other types of respiratory or gastroenterological problems? And there's opportunities because we are in a golden era right now for audio AI. We are entering into this phase where now our devices are able to understand our voices a little bit better, and they're able to translate the sound, and they're able to remove the noise from the sound. So why not use it for crying? So let me show you one of my demos on the next slide. So before we hop into a video, I'll describe a little bit about the context of what you're seeing here. So on the left is cry, and on the right is cough. So what we've done is we have taken cry audio from many children, and we have trained a neural network that is responsible for kind of reducing the dimensionality of the cry into just 2 dimensions. These are in a high dimensional space, so the dimensions just are important in that one point that's closer to another point just means that it's more closely related in terms of audio linguistic space. And so what we're doing is we're using this as a way to start collecting and quickly going through data for a participant that has, for example, colic. With colic, we might say, oh, this participant has been, the parent's reporting that they've been fussing for 4 hours every single day. Okay, well, how would we actually measure that as opposed to interview the parent? So the opportunity here is to record the entire day. We have a little recorder, goes inside a pocket in the onesie, and it records the entire day in audio. Well, now you have 24 hours of audio to review. So that's an incredible task. And especially if you have, for example, 60 participants in one of these studies, and you have 24 hours a day, and you're doing maybe 2 weeks of recording, you're looking at years, if not, like you're creating an incredibly large amount of work. So we need tools that allow us to pare down the data, remove all the noise, remove all the background noise, TV noise, adult noise, and only focus on when the child is making noises, and to regress and identify how much of that noise is in the fuss category, the cry category, or the laughter category. I mean, laughter could be an outcome as well. So what we're going to show you now is when you click on these individual points, this is what the machine learning algorithm was able to kind of cluster out and spread across. So when we click on different clusters, we should hear different types of noises. And then we'll show the one for cough right after. So go ahead, Ryan, and you can hit play on that video. So at the top there is a very different cry than the bottom.
Le va a llegar. Trarlo.
So you'll notice in these videos, they're not all coughs. So we make a AI algorithm, and with our partners, we have an AI algorithm that's listening for coughs and trying to find coughs, but it's not always right. And we want to make sure that's right. That's where I think there's a great opportunity in collaborating between artificial intelligence algorithms and machine learning algorithms and human annotators in the loop. So there's an opportunity here to be able to say, oh, that's not a cough. That's like a ringtone. That's someone's phone going off. It was a false positive. Or that's actually a cry. That's not a cough at all. And being able to cluster what is important to us allows us to draw a circle and say, when I hear more of that noise, that's the indicator I'm interested in. And like Jaya mentioned earlier, there are patients who are nonverbal, and there often are noises they make that we would like to know how often that happens. Sometimes the only way that a patient who's nonverbal can communicate their pain is through that noise. And so if we could say, oh, that's the noise we're looking for, take this recorder home, let's hear how many times that happens over the next few days now that we've titrated your medication in a different way. That creates an actual more quantitative endpoint we can take back to the clinic and take back into decision making. Now, of course, not all machine learning algorithms have the same amount of data. So I think it's important to note that it requires us to train on the individual person as much as on every baby that cries. So some babies just cry differently and they sound different. And you can imagine things about the way their anatomy completely changes the way the noises sound. So a child with cleft palate may have different resonance in the way that they cry versus another child that doesn't. So it's important for us to understand that we can't just take a model that works for one baby and immediately use it for another one, but we're creating the tools that allow us to slowly build that up over time. And so now we've talked a lot about like what can you hear and how can you turn that into a clinical indicator, but also what can you see? And one of the challenges that we've had is that in running clinical trials, we frequently ask patients and participants to take pictures of their skin or of their face or of their eyes, for rashes, wrinkles, acne, injuries, and post-operative scarring. And we want to better understand things like stool consistency as well. Can you take a picture and actually send that to a rater, and then the rater is responsible for interpreting it as opposed to just the participant? Of course, we still ask the participant so we understand that difference. But at the end of the day, when we see an underexposed photo, or photo that is of the ground, or it's not even the stool, it's just their countertop, we just lost a data point. And if you're a company trying to build the next device to make that child feel healthier, or you're working on a surgery, you want to understand the pain or the outcomes, that missing data point means you might have to enroll another participant, or you've lost the opportunity to truly understand their patient journey. So taking a good picture is something that actually phones are becoming fantastic at. The cameras are getting high quality, the algorithms are getting incredible. So we decided, what if we wanted to take a picture of kind of like a selfie app to support someone in taking a consistent set of angles across the head? And so we have created this app that allows you, a parent, to take pictures of the child's face at specific angles, and actually cuts off the top and bottom. And we can even cut off other parts of the picture so that the raters only see the important information. So for example, if we were only looking at the right eye of a child, we could actually take the picture from multiple angles, crop just the eye, and now the rater is no longer looking at many pictures of the backgrounds, getting distracted by bias. They're actually seeing just what they need. And there's opportunities there to create better environments for the annotators as well. On this next slide, you'll see some of the features that we were able to get out of the box. So there are incredible algorithms that we didn't even have to train. We just they were open source. We were able to take those models and manipulate them so they could actually extract the features of the face that we were interested in on a cell phone directly. So we could extract the lighting, the pose, identify the facial expression, so you're not squinting or moving your eyes and changing the position of your skin too much, and also confirm the identity and make sure the person you're taking the picture of is the right person. And you can see on the right, we're actually able to extract just the lips or just the part of the photo that we're interested in for different types of clinical trials. And so really what I like to think of this as is an AI algorithm that's supporting us in collecting the right data and helping the patient avoid a missed data point. And on this final slide, let's switch to the last one. Yep. So we have an AI-assisted diaper stool capture prototype. And the idea there is that, like Jaya was mentioning, sometimes color is more important than the consistency. It depends on what you're looking for. But of course, if you're in a dimly lit room or if the room has a big red wall next to it, it could be putting colors onto the picture and the rater might be seeing something that maybe the parent wouldn't see. So we want to make sure that we're taking the best possible picture and creating a good frame of reference. So we've included a little small color reference guide. So if we could send this little guide to the parent, that could be on the changing table, and as they take pictures of the stool, if that happens to be in the picture, we can very quickly identify a reference. And so there's an opportunity to kind of scan a stool, not just talk about, oh, it's a 7 or oh, it's a 6. So I think we're starting to explore that space of how do we best digitalize a clinical outcome and bring it to a rater or to a clinical consensus, and then really take these things to the next level. And so across both, what we're seeing and what we're hearing in the AI we're building, we're no longer relying on the parent to make a clinical observation. We are now working with the parent and working with the devices themselves to create better outcomes.
Brian, that's just been great, and I think these are terrific developments, and also they're very exciting aspirations because we're not quite there yet with everything. We are running a little late, so we'll quickly go to Ryan and polling question number 3, and then we'll have time for some Q&A. So still have a couple minutes to put that in.
Excellent. Thank you very much, Holly. Yes, before we move on to the Q&A, we've got one more poll question for audiences. This one is asking, what is currently the biggest impediment to deploying digital tools in your clinical trials? Your answer options this time around are security and privacy, patient ability to use digital tools, need for more tailored digital tools, data integration, or regulatory concerns. Give everyone a couple seconds to submit your vote. We greatly appreciate you taking the time to consider. Again, the question being, what is currently the biggest impediment to deploying digital tools in your clinical trials? We've got most of the votes in. Let's go ahead and take a look at our results for this one this time. 30% of you for regulatory concerns, 33% for need for more tailored digital tools, and then coming in at 90% security and privacy, 11% at patient ability to use the tools, and then 7% for data integration. Again, thank you very much. Holly, it's back to you.
Okay, thanks very much. So let's get to this very quickly. You can see the contacts for the speakers, and it'll be in the recording if you like. And something very exciting right off the bat here that we can hook up a little bit later. There's an individual who's talking about a device for autism symptoms and is very interested, Brian, in talking to you later on. So we will connect back to that person shortly. Let me get on to another audience question here. How can you normalize data from a child who is in a long-term study? I think they call out cognition as an example, I believe, to experience that cognition increases over time. So can you compare to baseline? Should you progressively change to other versions? For example, PEDS, QOL. I think, Susan, you might be the best person to answer this. Brian might want to throw in his 2 cents, but let's keep these short so we can have some more questions.
Sure, thank you, Holly. I mean, certainly, like you mentioned, that longitudinal data is critical. And, you know, there are periods in time where the assessments can work over many years, but I think to your point, at some point, you cross out of those assessments and you need to get to new assessments and you basically lose your baseline or your normalcy. I think the other thing that I would like to recognize, you know, autism was just brought up as an example, so I will go there. To my opinion, the tools out there for measuring autism and cognition are really horrible, outdated tools. There are things that are just textbooks like the Vineland scale. And then if you take a look at a lot of these tools, they're so outdated. You know, you're having children take a look at pictures of rotary phones and TV screens with antennas and things that, I mean, this is how old and bad pediatric tools are. I've had my son through many standardized assessments myself, and I sit there and I think, that's not his score. He's never, those things haven't existed in the last 40 years. How could he possibly know this? And so I get very frustrated myself as a parent when I look at the inadequacy of the tools and the equipment that we're using to assess cognition, autism, and things of this nature. And that is why, you know, a lot of my life is dedicated to and having such a passion for developing novel tools. We need to take a look at your study. We need to see what's critical to your endpoints and outcomes and what type of cognition are you measuring. Get rid of all the questions that don't matter, that are just going to frustrate the child in your care, and focus on that which is critical to a reliable cognitive assessment that can progress over time and as a function of age. I'll stop there.
2 cents, Brian?
I think that's a fantastic answer. I don't think I have anything to add to that.
All right, then let me throw another question your way. Someone wants to know if you are working on any digital tools to improve the accuracy in diet reporting. There's a big challenge. Do you care to address that one?
Sure. So you're saying diet reporting?
Right.
Right. So I think, yeah, we're inputs and outputs. I think we have a big, heavy look at the outputs. Right now, we don't have any active research programs right now around monitoring the inputs. But Jaya, how do you typically ask your patients to track what they're feeding their child, or how that changes over time?
It's a great question, and it's a challenge in the pediatric world. And if we want to track food allergies, on what types of food does that particular allergen reside in? There's so many hidden ways that, for example, wheat intolerance, which is very, very common. Did you know that there is a wheat additive in your toothpaste, in your lipstick? In certain drug formulations, which we don't tend to think of. So these are, it's very hard. So having a tool that you can, take a picture of that, and that can let you know if the food of interest isn't there or not. The second part is when we're looking at calorie counts for children who are not growing very well, and we want to assess how much are they able to get in and what types of foods they're able to get in. Recall is very, very fraught with problems. So being able to take a before and after picture of their meal tray, and then looking at a quick AI way to assess calories rather than somebody manually looking to see, is that half a cup, is this? So those tools are very helpful. I hope that answered at least a couple of points of how we would like to capture data.
Yeah, I think that's helpful. Thank you, Jaya. Susan, here's one you might want to field. How might the lessons you are learning in pediatric GI trials apply to other therapeutic areas?
Sure. Well, I think Brian touched on a number of those. Certainly when we think about the extension of these dermatology, and Jaya showed some fantastic examples of this, that space, as rashes. And now, especially that we're living in an era where vaccines and boosters are going to be with us for the foreseeable future. Long-term follow-up, injection site reactions, so easy to, right? if someone, over time, it's hard. You lose them to follow-up. But if it's as simple as taking a photo and that photo uploads to a cloud and either is looked at by a central expert, or to Brian's point, in the future we have AI doing all of this work across tens of thousands of photos, even better. And then certainly, I know work Brian didn't have a chance to talk about today, but both, we're very interested in movement disorders and how we can monitor those from the home perspective by doing short video clips that can then be, again, rated by central experts, or eventually someday we'll be at the point where AI can do all of these things. I think it's really important to think about when we talk about advancements in the pediatric space, there are three things that we can do. One is I personally have participated in the development and validation over 265 different novel EPROs. So what does that mean? Build better mousetraps, build better tools, the fundamentals. And then certainly the digitization of them increases compliance, adherence, engagement, and so on. And then having the technology to facilitate and remind is a way to do that. The second thing is, and you saw some examples of this today, if we can get a parent to send us an image or a video clip, there are many of those cases where we don't have to wait for AI to come online. Experts like Jaya can take a look at hundreds of those images and very quickly provide ratings in a standardized fashion. So a combination of the parent contributing an image coupled with a central expert is a technology that we deploy today. And then to Brian's point, eventually those gigantic data sets, and they're already there, we just don't know what to do with them quite yet. But they're coming along where no human, Jaya's whole team and more would not have the time to process. And we need machines, and we need tools, and we need reliable algorithms so that we can detect signal among all of our capabilities. So there we're at a place where our capabilities to record have outpaced our capabilities to analyze. But thankfully, we have people like Brian who are so good at lining up and figuring out how to bring signal out of those voluminous mega data sets.
That sounds good. Brian, did you have anything you wanted to ask briefly? We're almost to the end, although I think we might run over just a couple of minutes. Did you want to add any more to Susan's remarks there, or would you like another question?
I think what Susan's speaking to is this path that we take towards incorporating AI and machine learning algorithms in a more intrinsic way of clinical trials. That, of course, is going to lead to a complete revolution in the way we take our devices to market and the way that we evaluate new therapeutics. So it's going to be a very exciting and interesting path for the way that we're going to start changing clinical trials and the foundations of how we interpret data. And one of the simplest examples of that is if I take an hour-long recording of a baby and hand it to every person in this webinar and ask you all, say, how many minutes of crying was there? All of us have a different response. And I think there's an important note that a lot of these ePro are not measuring the observation, they're measuring the perception of crying or the perception of stool consistency, not the actual stool consistency. So it's going to be really fascinating to see how those change over time.
Well, that's interesting because the perception issue kind of leads into another question that we have here that I think you might be able to answer, Brian, or at least start to address. How can we improve parent report of physician-diagnosed conditions while avoiding the tendency of parents to self-diagnose, which is something that Susan brought up earlier. How do you think we can approach that question?
So one of the challenges we have for an existing trial right now is around colic. And of course, we want to make sure that there is source data verification, that when a participant enters our study, they actually have the diagnosis from their clinician or from a certified clinician, so they don't come in saying, oh, I think I have that problem, I should be a part of that clinical study. I think that we need to start incorporating new screening methods that go a little deeper, go beyond just, oh, you said you had that problem and welcome to the study, or you said you felt like you had this, this condition, so now here's your first medication for the day. I think that's going to be a great screening challenge and making sure that you're incorporating new tools into that screening directly. For one of our studies right now, we are using an AI-generated approach to extract how many minutes of crying there is in the audio, as well as asking ePro for the audio for colic. And we're having a clinician review both to make sure that they're at least pretty close before they say, okay, you're in the study. So I think there's a lot of opportunities to look for disparities in the data, and of course, still leverage the state of the art of patient-reported outcomes, but also check too.
Great, great. All right, we're going to wrap up with one last question. I think, Jaya, this one is directed to you. What are the possible implications for applying the stuff that we've been talking about that's shared between sort of your sort of practice and the clinical trials to this stuff actually morphing into something that can be standard of care? I'd love to hear your opinion on that myself.
Great question. I think the applications are unlimited. In my daily practice, there are so many places, even within the hospital, as we were talking about, standardizing what the nurses observe. It is hard to have somebody 24 hours in the child's room. And just as, we all talk about how as soon as the doctor walks in, you can't remember your questions, or you can't, that's the only time the child doesn't have a symptom. Doesn't mean that's not true, but how do you capture that data when a clinician or somebody who can analyze that data is there? So tools will help. Tools that take out the human recall or human interpretation and provide data across time to be analyzed is very important. It's helpful for training new younger generations. I mean, there's the saying, right? The old doctor is the one that knows the most because they've had the years of experience. How do you condense that experience into a tool that younger trainees can leverage off of in information over time? So we have that ability with AI right now. If AI can tell me where to buy my size clothing, I hope it can tell us what type of treatment or data that we can gather to help health.
Oh, that's a wonderful way to conclude the questions. I'm sorry we didn't have time to get to all of them. We'll see if there isn't some way perhaps we can respond to you personally. But thank you everyone for attending. This has been really very interesting, hopefully as interesting for you all as it is for us. And I know we need to get back to Ryan. He has some wrap up for today. But again, thank you everyone for coming, and I give it back to Ryan to close us out.
Well, thank you. Thank you very much for that wonderfully insightful presentation and excellent Q&A session. However, we are out of time here today. As you said, though, if we couldn't get to all the questions, the team at Optio Health will follow up with you after the webinar. But if you have further questions, you can direct them to the email addresses that are up on your screen. I also want to thank everyone in attendance for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking all of our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.
Thank you.
Thank you.
Thank you, everyone.